Assalamu alaikum and good morning. Today I'm going to talk about vaccine as part one, and Dr. Crystal will be talking about issues related to immunization as part two. Management of disease can involve either one of the following therapeutic procedures, for example, surgery, uh, drugs, antibiotics, antivirals, or gene therapy. And also, there are other measurements or measures, such as prophylactic measures, which are used to prevent earlier disease from occurring. Vaccination against pathogenic microorganisms is one of the most important advances in the history of medicine. And during the last 200 years, the vaccination programs has been able to control or eradicate 10 major diseases, at least in the world. Mammals are protected by a variety of mechanisms. Some of these mechanisms are non-specific, while others are highly specific. Among the non-specific, for example, is the skin, and among other highly specific is the response of antibodies against antigens or foreign material entering into our bodies. So what is active and passive immunity? Now, naturally acquired active immunity usually occurs in a person when he is exposed to live pathogens, and he develops a disease and becomes immune as a result of the primary immune response. Artificially acquired active immunity can be induced by a vaccine, which is a substance that contains antigens, and a vaccine usually stimulates the response against the antigen without causing symptoms of the disease. Again, passive immunity also occurs in two different ways, either artificially, where this is usually a short-term immunization, which is achieved by injecting antibodies, such as gamma globulin, and usually this is taken from one person to another, or naturally through passive immunity during pregnancy, in which antibodies are passed from the maternal to the fetal bloodstream. So what is the goal of immunization? Usually immunization can partially or completely prevent the illness which is caused by a microorganism. And by preventing illness, immunization can protect you from acute effect of a disease, complications, long-term disability, and other issues. And usually immunization are widely used. If they are widely used, then the spread of a disease within the population can be prevented. And this is just the aim at the moment of trying to find a suitable vaccine, for example, against coronavirus, that is to help in controlling the disease within the population and prevents the outbreak and reduce healthcare management. Now, the word vaccine comes from the Latin word for cow or vacca, which is a product of weakened or killed microorganism, whether it's a bacterium or a virus, given for the prevention or treatment of infectious disease. Historically, when we think when was vaccines introduced, we find that if we go back 3,000 years before Christ, that means that nearly 5,000 years ago, there was evidence of people sniffing powdered smallpox crust in Egypt. And then in 2000 BC, sniffing of smallpox crust was also observed in China. The Turks introduced the variolation, something similar to inoculation, in 1500 AD. And in 1700 AD, the variolation was introduced in UK and later to USA. Now, early opposition of the use of cowpox was based on some idea that people thought that the agent, which is taken from cows, might induce cow-like characteristics in humans. So that's why people really rejected the idea of the use of cowpox at that time, or crust at that time. So earlier we mentioned that the Turks introduced variolation. And you know, at the time of the Ottoman Empire, there was relations between different countries, and the British ambassador's wife in Turkey saw that variolation of her son really helped him in being prevented from dying because of smallpox. And at that time, she thought that she would be a patriot enough to bring this invention into use to England. Later on, several years in 1796, there was an English doctor whose name was Edward Jenner, and he was experiencing and experimenting with variolation. However, he discovered that there are other ways of handling protection, and among them is vaccination, and that is by using vaccinia virus, the agent of cowpox. He found that those ladies who usually worked on milking the cows did not really get infected with smallpox. And when he checked, he looked and saw that they had some sort of vesicles on their skin, which he thought that they might have gained this from during milking the cow, which was infected with or had these vesicles on their skin, and could be some sort of cowpox. So therefore, he took few drops of the fluid from the skin of a sore woman and who had cowpox, and injected that fluid into the arm of a healthy young boy who has never had cowpox or smallpox. And after a few weeks, he challenged the boy with smallpox sore, however, the boy remained free of smallpox. Now, what are vaccines made of? Usually the vaccine is made from proteins that are called antigens, and those are known to stimulate the immune response. Those are isolated or produced from microorganisms that causes the disease. And then the vaccine is injected into the bloodstream, and hence the immune system will start developing antibodies. Now, obviously, we cannot use live or virulent organisms as a vaccine, because that means that it will induce the disease that we are aiming to prevent. So therefore, we need to separate this effect of microorganisms that causes the disease, meaning that we need to isolate an organism or part of it that usually is known to cause the full-blown disease, and try to use the antigens that can induce the immune response. How can this be done? This can be done in many ways. Now, the strategies that have been adopted in the process of trying to find appropriate vaccine can range from either whole inactivated vaccines, live attenuated vaccines, live recombinant vaccines such as viral vectors that are utilized to transmit a piece of nucleic acid, virus-like particles, for example, subunit vaccines, synthetic peptides, meaning that you synthesize a protein, a small protein that is similar to the immunogenic protein that is required, or through the use of lipid nanoparticles that are encapsulated with messenger RNA. In this case, usually this is a synthetic messenger RNA that is delivered to the cells, and those messenger RNA are going to be expressed making new proteins or immunogenic proteins, or through the use of naked DNA vaccines. Those are just a piece of plasmid that contains promoter and different sequences that are needed to express a certain gene that is inserted into them. In this diagram, this represents the different strategies which we can use or utilize to make different types of vaccines. For example, in the middle, you can see the virus particle. The virus particle can be treated with certain chemicals that lead to killing the virus or inactivating the virus, which can be utilized further as a vaccine, or by attenuating the virus particle, meaning that you introduce certain mutations that affect the virus and cause it to become less virulent or attenuated, hence not causing a disease, and can be utilized to be used as a vaccine, or fractionating the virus into different components and using those protein components as immunogenic proteins to induce the immune system, or by synthesizing the units by making proteins that are immunogenic, or by making antibodies that usually have three-dimensional immunogenic regions. Again, what we can do is we can clone parts of the genome or genes into yeast and expressing that and making large amounts of certain proteins that can be taken and utilized, or by subcloning into virus vectors or DNA vectors, and those usually are utilized to express, again, the immunogenic proteins that induce the immune system. As I said earlier, during the last 200 years, there has been vaccines that have been able to control so many diseases, whether these are bacterial or viral or others. And as you can see from this list, which I'm not going to go through, that there are different diseases or different vaccines against those viruses or bacteria and so on. Now, one of the major obstacles that exist when making a vaccine is, first, very little of the envelope spike surface of the primary virus appears accessible for antibody binding. Hence, this is usually low antigenicity proteins, and probably that is due to the oligomerization of the constituent proteins and high degree of glycosylation of one of the proteins that really cause them to become low antigenic, or antigenicity, of low antigenicity. The other obstacle, or the second obstacle, is that mature oligomers constitute the spikes appear to stimulate only weak antibody response, low immunogenicity in this case. So these are one of the two major obstacles which we tend to really find when trying to make. So earlier we mentioned that there are different strategies to make vaccines. However, up to recently, we have been involved in two main types of vaccines: those that are live or attenuated, and those that are dead or killed vaccines. Now, those two vaccines, they have their positive and negative features. For example, in terms of dosage, with live or attenuated vaccine, usually the dosage is low in comparison to dead or killed vaccine. The number of doses in live vaccines usually is less or single in comparison to multiple doses required for dead vaccine. And in terms of the use of adjuvant, with live vaccine, you do not require adjuvant, and we'll be talking about adjuvants in a while. In comparison, dead vaccines, yes, you do need adjuvant in that case. The duration of immunity which is being observed is, in the case of live vaccines, is for many years, while in dead vaccines usually is less. Cell-mediated response usually is excellent, and this can vary for dead vaccines. Reversion to virulence, and what do we mean here is that, if you recall, we mentioned that live or attenuated vaccine can be achieved by introducing mutations. Now, this causes the attenuation. However, these mutations sometimes can revert to the wild type, hence causing a real disease. And this can be observed with live vaccines in comparison to dead vaccines. It's not possible. So altogether, the other issue which is really important is the safetiness of these vaccines. Both, as I said, they have their positives and negatives. For example, one of the potential problems with live vaccine is the under-attenuation. So therefore, it's very less mutated or very less attenuated to the extent that the immune system is not capable of making antibodies. As I said earlier, the mutation can revert to lead to virulence. Contamination of the preparations with cultured cells, those attenuated preparations usually are prepared as live and isolated from culture. So the culture cells can also be present in certain preparations. The preparation itself, sometimes it's not stable. And also they are heat liable because simply that, these are attenuated live, so therefore they can be affected by high temperature. And one of the issues here to remember is that those live vaccines should not be administered to immunocompromised people or pregnant patients. And as I said, the live vaccines and certain viruses can induce reactivation and can be oncogenic in certain cases. The same thing with the safety issues for incomplete inactivated vaccines. There is an increased risk of allergic reaction simply because you'll need large amounts of doses here, and this is going to involve large amount of antigen, and also present problems with the development of the vaccine, which can include failure to grow in large amounts in the lab, simply because some viruses are not that easy to grow in the lab. And sometimes the crude antigen preparations can give poor protection. For example, the key antigen is not identified, hence the protective immune response is going to be less. Now, viral variation is among the other possible obstacles that appear to present problems that can affect the vaccine that are being made, or the vaccines that are being made. So classical vaccine strategies based on attenuated live or whole vaccine has severe limitations, as we mentioned earlier, since those classical vaccines can prevent development of disease but do not prevent infection. And they allow a limited number but controlled replication of the pathogen at the portal of entry. Still, so you have replication which is occurring. And hence one might think, what is the features of an ideal vaccine? What do we look for in a modern vaccine? So one of the issues is the no risk of infection. The raised antibody against the native forms of the proteins, raised cytolytic T-cell response, raised long life responses, facilitate the use of a combination vaccine, good stability at low and high temperature, avoid the risk of permanent integration into the host genome, meaning that the high possibility of having the nucleic acid being integrated into the patient's DNA. And this is not acceptable in such situation. Therefore, the question which we ask ourselves is, what is an ideal vaccine now? Is it a DNA vaccine? Is it an adenovirus vector vaccine? Or is it the lipid nanoparticle encapsulated messenger RNA vaccine? We'll leave these later on, or the answer for these later on. Now, one of the issues which I wanted to mention is that when we prepare vaccines, there are certain constituents which are added, and those constituents are necessary to be added to the vaccine preparation. And usually we refer to them as additives. So those are very small amount, and they are very essential to ensure the safety and effectiveness of the vaccine. Among those additives is the adjuvant, which we just mentioned about it, and the use of adjuvant is to enhance the immune system response, such as aluminum gels or salts, antibiotics, which prevents bacterial and fungal growth, such as neomycin, streptomycin, stabilizers, maintain the effectiveness in face of adverse conditions, such as sulfites, monosodium glutamate, gelatin, preservatives to keep the multi-dose vials of vaccine sterile after they have been opened, such as formaldehyde or other chemicals. Among the vaccine additives, which we mentioned several times, is the adjuvant. Now, what are adjuvants? Adjuvants are those compounds that bind to the antigen in the vaccine. And usually they help in retaining the antigen at the site of injection, and also help in delivering the antigen to the lymph nodes where the immune responses to the antigen are initiated. The slow release of the antigen to the tissue around the injection site and improved delivery of antigen to lymph nodes can produce stronger antibody response than if the antigen was there without those adjuvants. Alum adjuvant also is known to be taken by cells such as macrophages and help these cells better present antigen to the lymphocytes. So making a safe vaccine is probably the most important issue when trying to prepare a vaccine. It usually takes a very long time to prepare a vaccine. In certain cases, it can go up to 10 years or more. Why? Because vaccines must be proven safe and effective before reaching the public. In order to have a vaccine licensed and given to the public, there are steps which really the vaccine has to undergo before reaching the licensed stage. Among these stages or tests is at the beginning is the laboratory tests, where the vaccine would be tested in the lab. And then after finding that the results are successful, the testing will be on animals and lab animals. Then when again this is going to be successful, it will go or undergo human clinical trials. And the human clinical trials involve three main phases: phase 1, phase 2, and phase 3. We will mention what are these phases for. Phase 1 usually helps in studying the safetiness and immunogenicity of the study. Uh, Usually the number of participants is limited to 1 to 20, 30 probably individuals. While in phase 2, mainly here the concern is trying to study the safety of the vaccine and the dose range that should be utilized in the study. Usually the number of participants here is around 100 or hundreds. In the last phase, phase 3, or the large, usually this is the large scale, again, the safetiness is very important and the efficacy of the vaccine is studied. Usually this can involve several thousands of people in the population, and all this is going to generate data which is going to be studied thoroughly in order to really evaluate the vaccine and help in licensing the vaccine for further usage among the population. In phase 3, we said the trial subjects is multi-center and several thousands of subjects are involved. The goal mainly is to provide data of safetiness and efficacy, and the efficacy usually is measured by the incidence rate of the vaccine group divided over the incidence rate of the control group. This again is taken into a formula which will give you the vaccine efficacy that will ensure that the vaccine has reached the best, let's say, efficacy that is to be administered to the population. For corona vaccine, as the latest statistics tells us, that there has been something like 105 vaccines under different stages of being tested. Some of them are still in the early stages at phase 1 or phase 2, and some have been authorized, and that means that they have been accepted for limited usage, and some have been approved for full use, as the ones which we have been taking, such as Moderna, such as Pfizer, and other types of vaccine, Johnson, and so on. And as you can see here, in the red square, there are 9 vaccines that have been abandoned, meaning that they have not been given the okay, or their results were not really encouraging to be used as a vaccine for people. Having said so, now this is a list of the leading vaccines which we know, and you can see there is the column which represents the developer and how this is made, or how it works, and the status of this vaccine. So, for example, Pfizer, it works on the basis of messenger RNA. The other types, Moderna is similar. Other types based on the vector, virus vector, adenovirus vector, such as Oxford AstraZeneca and CanSino and Johnson & Johnson, and so on. And you can see the Chinese vaccine, where there is it's based on inactivated viruses, and has been approved in some of the countries altogether. So this is just to give you an idea that those that have been utilized during the recent months. This here diagram really tells us, if you remember, we said that preparing a vaccine usually takes something like 10 years. Now, what is the situation with the COVID vaccine development? As you can see here, in comparison between the classical or typical type of development and the development of corona vaccine, the time has been shortened to nearly 2 years instead of having over 6 years. And all the trials have been packed, phase 1, phase 2, and the large number of people that have been used. All these have been utilized, aiming that within the next year, the vaccine will be approved and distributed among the population. Having said so, this is with regard to coronavirus. There are, of course, other viruses. Now, there is a recommendation that people should take influenza vaccine so that at least they are not affected by influenza that are distributed among the population. And usually there is a vaccine that is prepared every year against influenza, and those vaccines, usually there are two types. Again, there is the quadrivalent and there is the trivalent. The quadrivalent usually involves more than one virus, so usually it has four different types of viruses, while with the trivalent usually there are 3 types of viruses. So it's a mix of viruses depending on whether the virus, or what are the viruses that are circulating in the northern hemisphere or those that are circulating in the southern hemisphere of the world. Now, other than that, there is also the polio vaccine, for example, and those are different types of vaccine. There is the monovalent, bivalent, and then there is the inactivated polio vaccine. We know that one of the aims of the vaccine here in this case is to eradicate polio from the world. Polio is a very old disease, and we hope that within the next coming years we will be able to eradicate. But if we tend to look at the amount of effort that has been used to eradicate the virus, we will really have an idea how things are going to be dealt with coronavirus. So I think that coronavirus will not be disappearing in a day or two. It will take quite a long time really for the virus to be eradicated, similar to what we see with polio virus. Here, this is one of the latest distribution of different infections with polio, and there is the wild type polio, and there is the polio which is generated by the vaccine itself. You remember we said that there is a high possibility of reversion of the virus from the attenuated to the wild type. So this is what really happens in certain parts of the world, where the virus really becomes wild type, hence causing big issues of infection. Again, the red area represents the place where the wild type is concentrated, especially in Afghanistan and Pakistan. Now, success and failure of vaccines can vary. We keep hearing vaccines and we think that the vaccines are always successful. but this is not the case always. If we go to the story of RotaShield, we'll find that RotaShield is a vaccine which was made against rotavirus during 1998, and it was used for, as I said, for rotavirus that infect children. And what was found is that there was a strong association between RotaShield and bowel obstruction among some infants during the first 1 or 2 weeks following vaccination. And therefore, this vaccine was withdrawn from the market, although it has gone or undergone phase 1, phase 2, phase 3 of development. However, still, this did not really help in the success of that vaccine. At the moment, there is another vaccine, Rotrex, and this was developed in 2006. It's an oral liquid vaccine that provides protection against so many rotavirus serotypes, and it seems that there are no bowel obstruction problems associated with this vaccine. Hence, the vaccine, the new vaccine is successful in comparison to that old vaccine. For coronavirus vaccine, that still needs to be seen. How those vaccines, which are at the moment being under experimentation and development, we need still to see how effective they are and how successful they are going to be in the future. One of the issues which we probably did not mention is the type of administration of a vaccine. Now, probably one day we will see even vaccine being administered through our food, or maybe eaten at the dinner table, or be given through bananas, for example. And simply this can be achieved by introducing certain genes into the food which we eat and so on, or by applying skin patches, or by using nose administration, or gene gun, or spraying. And as a matter of fact, spraying is even utilized now for H1N1 or influenza vaccine. So the utilization or the administration of the vaccines will, I think, will develop again with time, and we will see better ways and means of administering vaccines. In conclusion, vaccines are very important in maintaining public health. Usually they should be safe, cost-effective, and they are the efficient way to prevent sickness and death from infectious disease. I think this is where I'm going to stop, the end of the part 1 story. If you have any questions, please do not hesitate to contact me with your questions, and I'll do my best to answer them. The part 2 is the talk by Dr. Crystal will be very short in terms of trying to explain the immune system, how this reacts to the vaccine. Thank you very much, and I hope to hear from you.
So in the first part of the lecture, you have learned different types of vaccines that are currently available. In this remaining part, we're going to take a look at how vaccines induce immune response. Now, vaccines are made from immunogenic components of microorganisms, and these immunogenic components are usually virulent factors of the pathogen, and they usually contain proteins. And you have also learned in this lecture that vaccines can be made from the entire organism, whether it's whether they are inactivated or attenuated. And some vaccines are smaller components from that organism. So when it is a component of an organism, that component can be either isolated or they can be synthesized using genetic engineering. Now, occasionally, immunogenic components do not contain protein. So, for example, bacteria, its virulent factor, it contains only carbohydrate and lipid, and it does not contain protein. In that situation, the virulent factors must be attached to proteins in order for them to be immunogenic, and we will take a look at why that is in a minute. Once vaccines are produced, vaccines can be administered in 3 different routes. They can be subcutaneous, just slightly under the skin, or they can be intramuscular, injection into the muscle, and that's usually the muscles of the upper arm. And some vaccines can be taken orally. Once the vaccine is administered, say in this case of intramuscular injection of a vaccine, then that vaccine over time slowly gets into the secondary lymphoid tissues, into the draining lymph node in this case, and the immune response to vaccine occurs in that draining lymph node. In other words, vaccine, regardless the route of administration of the vaccine, the immune response occurs in the secondary lymphoid tissues because, as you already know, that's where vast majority of T cells and B cells reside. So I did mention that good immunogens, or in order for vaccines to induce immune response, that immunogen must be attached to protein. So either the immunogenic component already contains protein, or if the virulent factor does not contain protein, they must be attached to a protein component that is not toxic to the host to induce good immune response. In other words, for the vaccines to be efficacious. The reason for that is something that you already know. Protein is necessary to induce and activate T cells, right? So you already know that an antigen can be taken up by antigen-presenting cells like macrophage, and protein component is degraded into peptides, and the peptides are presented in the context of MHC class II, in this case, on the antigen-presenting cell, which would be recognized by the T cell receptor expressed by T cells, right? And similarly, even when B cells recognize the antigen through B cell receptor, they uptake the antigen, gets into the endosome. It's the protein component that gets degraded and gets put onto the MHC class II, and therefore now the peptide from the protein is present in the context of MHC class II, which can be recognized by helper T cells. So without protein component, T cells cannot recognize any antigen. Therefore, vaccines, effective vaccines, must contain protein component in order to be able to activate T cells. Now let's take a look at immune response to vaccines. Once the vaccine is administered, that vaccine should be able to activate CD4 T cells, and CD4 positive T cells differentiated into helper T cell subsets, and they produce cytokines which promote effective humoral immune response. More importantly, as they respond to the vaccine, the generation of memory CD4 positive T cells result. And it is important to generate memory T cell subsets that are Th1, Th2, Th17, depending on which organism is being vaccinated against. Of course, if it is for intracellular pathogens, then activation of CD8 positive T cells and generation of memory cytotoxic T cells would be very important. And this would be quite effective when using live vaccines. Even the dead vaccines would generate activated CD8 positive T cells and memory cytotoxic T cells if the vaccine is against intracellular pathogens. And the other arm of adaptive immunity would be humoral immunity. So B cells would interact with vaccines and be activated. And these activated B cells then undergo the differentiation stage to become plasma cells to produce antibodies. Now, under the influence of cytokines produced by helper T cells, then they can switch from IgM to IgG and IgA. And these antibodies are very important for prevention of future entry of the pathogen that the vaccine is against. And of course, the key of the vaccination is generation of memory cells and therefore generation of memory B cells, as well as long-lived plasma cells that come from the initial response during the vaccination would be the key factor or key result of vaccination. Now, what I would like to do is to compare different features of immunization and features that are related to immunization, looking at live vaccine versus dead vaccines. Now, when I say dead vaccines, this includes heat-killed vaccine of an entire organism, as well as a component of an organism that are either isolated and purified or genetically synthesized. So in terms of the dose required for effective immunization, live vaccines require low dose relatively in comparison to dead vaccine, which requires higher dose than live vaccines. And in addition, number of doses, in other words, number of shots that one has to get is far lower in live vaccine, where single shot would be effective in inducing immunity, as opposed to dead vaccines would require multiple doses. So there would be initial dose number 1, and then a month later, another dose, and then 6 months later, another dose, which we call booster shots. So it requires multiple shots. And in terms of the need for adjuvant, live vaccines would not require adjuvant, while dead vaccines would require adjuvant. Now, the function of adjuvant, or the adjuvant, is different types of substance that would aid antigen or the vaccines to be held or to be retained in the site of injection for a longer period of time, and then slowly releases the vaccine, so that the small amount of vaccines would continuously stimulate the immune system, in particular cells of the adaptive immunity. So for dead vaccine, it requires adjuvant would be very, very helpful in terms of enhancing the immune response. Now, the duration of immunity after vaccination, if live vaccines are used, they last for many, many years, and dead vaccines would be less than live vaccines. Antibody response, both live and dead vaccines would induce IgM, IgG, and IgA, provided that dead vaccines include protein component, as I have mentioned. Cell-mediated immune response inducing T cells. The live vaccines are really good at inducing cell-mediated immunity, while dead vaccines, it can be very good, but the efficiency of inducing T cell varies. Again, because of the type of protein present, the amount of protein used, etc. Now, of course, one caution that one has to look at is the possibility of reversion to virulent pathogen itself, where live vaccine, it is possible, even though they are attenuated, where dead vaccines, that it is not possible. Okay. Now, finally, this is a summary slide for you in that when we have vaccines that are efficacious, the goal is to induce memory. Now, when we talk about memory, we're basically looking at the secondary response that immune response can provide. So initial vaccination would be primary response. The booster shots, the second shot, third shot, would to stimulate secondary immune response when the dead vaccines are used. So in this graph that we're looking at antibody level as a measurement of efficaciousness of the vaccine, and that is on the y-axis, and then the time of the vaccination and immune response that can be observed. So initial dose, one would primarily produce IgM, and there would be somewhat of a switching to IgG a bit later, indicating that T cells have been stimulated. But when there is memory response, obviously IgM is produced because even in the second injection, naive cells are stimulated in addition to memory cells, but there are memory cell responding. So in this case, it is the B cell, memory B cell response being measured, lots of IgG and lasting long time, and therefore providing ample protection to the host. So I will stop right here. Okay, if you have any questions, please send it in through the forum, or bring the question to the tutorial that we will have later. Thank you for your attention, and you have a great day.